people who come late. So please start.
Good morning to one and all. Greetings from Srishti and Honeybee Network. This is a BIIS-11 workshop. We are coming to an end. We have started on 7th March 2022, and we are here on the final day of the workshop. Good morning to one and all. I welcome Professor and Honeybee Network, Professor Anil Gupta, to deliver the welcome address. Professor?
Yes. Good morning, friends. We have a very distinguished friend, colleague, an eminent scientist with us, the former director of CIMAP, Central Institute of Medicine and Aromatic Plants, CSIR lab in Lucknow, director of Institute of Science at Banaras Hindu University, Varanasi, and a very accomplished scholar, DC Bose Fellow, and numerous other distinctions that he has received. So I will request him to inspire you. But before that, let me mention that what our purpose was for this BIIS and how this program is being now scaled up in the next phase to lead to what is called as Bharat Ke Sitare. So, the hope is that in future we would have not just 35, 40, but 1,000 students per program. And there'll be 4 such programs in a year. And from those 1,000, we will select around 100 who will get exposure to offline experience, some of them in different labs. And then around 20 or 30 would get even more detailed immersion. Finally, we will select 10 who do very good projects and translate them in the field, talk to the communities and ensure that either they solve problem with the community, co-creation of solutions, or they also try to disseminate an existing solution in a participative manner so that communities benefit. So essentially, the idea is that from induction in science, they go to society and translate the scientific understanding into societal change, societal impact. This is the purpose of the new phase. 10 of you will be selected in future as Bharat Ke Sitare, and they will be given the award by Honorable President. Now, the reason for recognizing the students in this manner as Bharat Ke Sitare is that we believe that some of you will indeed be the future leaders of our science, of not just science, but translational science, science that makes impact at the societal level. And we are very hopeful that you will bring about change, you will inspire your own peers. And as you know that many of you were also trying to link knowledge from the elders, from the community seat elders. You try to bring knowledge of the service provider at your home. Some of you did home experiments. So our hope is that it will be possible for you to integrate the knowledge of our communities, knowledge of our elders, knowledge of our society, and knowledge of hardcore science. All of this is possible with determination, dedication, and devotion. I must also say that during this program, some of you did very good assignments. We are very happy with them. Some of you already developed your project, which you will present today. A few of you have been selected today among those who had submitted the projects to present, and we will get the feedback of Dr. Anil Tripathi. It is very important to mention that when scholars like Professor Tripathi come and talk to you, it is not only a privilege, but also an opportunity for you to build linkages with outstanding mentors of our country. It is possible that some of you might need more detailed inputs into your project. And I have no doubt that if your questions are worthy of attention and worthy of in-depth analysis, then senior mentors, senior leaders of our science in our country, such as Professor Tripathi, would be very happy to guide you as and when situation arises. So you are in that sense building a relationship with one of the most senior mentors of our country and who has accomplished and in some sense made a huge impact, because when he was director of CIMAP, I had the opportunity to go there and see how good science was translated into good impact, societal impact. So I'm extremely happy that you got an opportunity to meet him today. But you also met a lot of distinguished scientists during the program from various disciplines, from CSIR labs, from DBT labs, from University of Sciences, from IITs, from ISER, from various incubators. Why did we bring such a diverse experience to you? We brought such diverse experience to you because we wanted some of you to aspire for more impactful careers in future. Some of you might become entrepreneurs. Some of you might become applicant for Gyati Award, Gandhian Young Technological Innovation Award, which is 15 lakh rupees to 15 scholars as a student for 2 years. And in big grant, you get 50 lakh rupees. Sometimes when even you don't have a proof of concept, just for an idea. And that is another support that Biotechnology Industry Research Assistance Council, an autonomous body of TPT, which has partnered with Srishti in creating this platform of BIIS, they provide you that opportunity. So the purpose is, and I hope Shilpi has joined us, so she would also be, has she joined, Amar? Just checking the participant. So the colleagues from BIREC are very keen that all of you remain in touch with the program team at Srishti, at Honeybee Network, and try to pursue bigger challenges in your life. So I would also like to mention that there were different assignments that most of you have done. Some of you have not done. I will persuade you to do them, complete them. Assignment about knowledge of elders, knowledge of your community, knowledge, doing literature review on grassroots innovation practices, and of course, protocols. I realize that some of you are very busy with exams, so you may be able to, you need another week. We will be happy to provide you one more week. But remember that selection of the final 10 who will get 1 lakh each will depend upon the rigor with which you submit the assignments and the proposal. So I'm hoping that none of you will spare any effort to compete for the top slot and therefore be able to contribute. So I really appreciate this opportunity to have met all of you. And I hope that you also found your program useful. We will send you a feedback form so that you can send us session by session your advice, your suggestion, your reaction to the content delivery and its relevance to your future expansion of your vision, so that we can further improve this program. As I told you, this program is going to be scaled up. This program is going to get many more students. This program is going to get much more exposure to the entrepreneurial process also, and many more institutions are likely to be involved in this, though it will be anchored at Srishti itself. So with these words, I once again compliment you for having completed this program, but formal completion will be when you submit all the assignments, all the requirements, and only those who complete all the requirements would get the certificate as is expected. That is the protocol, that is the discipline of any program. So I hope that you will not hesitate, you will not delay your submission of the all requirements that are necessary. Amar, I will now, if you want to show something about Dr. Tripathi, then you can bring the profile of his. But colleagues, let me tell you that Professor Anil Tripathi, before we are going to get some presentation, Amar, before we request him.
Yes, Professor, we have.
So why don't we have a brief presentation of 5 or 6 projects so that Professor Tripathi can get an idea as to what have these students, undergraduate students have tried. Sure, and then during his talk, he might also like to give us some little feedback as to how can they do better.
Okay.
Thank you.
Kirti, are you ready? Can you start?
Yes, Professor. Good morning to all of you. Am I audible?
Yes, Kirti.
Yes.
Good morning, all of you. I'll be sharing my screen. Is it visible?
Yes.
So once again, very good morning to all of you, all the dignitaries and my colleagues during this program. Today I'm here to present my project proposal, which is on the theme evaluation of the Phytophthora aurea, that is money plant, for its antimicrobial action. Starting with the introduction of the plant, money plant is grown in almost every Indian house. We see it here, there, growing enormously without any special care. And we don't consider money plant as a medicinal plant in India.
Kirti, excuse me, your screen is very flickering. Yeah.
It is clear from my side, Professor. If it's possible, can you share the screen?
Please carry on. Make it brief, make it very brief, because there are presentation. We have only half an hour for that. Just start, continue. Please continue.
Yes.
So basically the idea behind the project is that I have planned to screen money plant for its antimicrobial action, because since childhood I have seen money plant growing in my house, in my society, neighborhood, but it never got any kind of fungal infection, microbial infection as generally plants get. So the idea of screening it for its antimicrobial action arises from there. I have already done methanolic extraction of the plant and planning to check this extract for its antimicrobial action against Candida albicans and E. coli. If I'll be having promising results, then I'll be trying to test it on further more pathogenic bacteria, because we'll be having a natural herbal and very easy growing cure for the antimicrobial substituent. This is the plan of work that I have planned. I have completed the extraction. Further is yet pending. If possible, I'll also be trying to elucidate the structure of this special compound which is responsible for the antimicrobial action. I have already having a review article published on the same plant with my mentors. That has been published last year in August about the whole kind of activities, because money plant is having almost 65 plus compounds which are secondary metabolites, and they all are having very significant actions. Even money plant is having nicotine absorption. It is, even NASA have proved that it absorbs nicotine from the surroundings, and it is having very calming action. It is used for not only that purpose, but also for cleaning the air. It is used in interior scapes. These are some mentioned references, and I am having almost 62 to 65 references for this project, as I have done a literature review already. So this is the proposal I have to submit, and that's all for it.
Okay.
Thank you, Kirti. Next, Rasi Verma.
Yes, sir.
I can start person.
Okay, sir.
Yes, Rasi, you can start.
Please start. Please start. There's no time to waste. Rashi, if she's not ready, then we should. Rashi, we can't hear you. तुम्हारा माइक में प्रॉब्लम है? चेक करो उसने माइक के साथ नहीं, तो किसी और को बोलो. Ask somebody else.
Pratima.
इसको remove कर दो.
Yes, sir.
Please remove this. Please remove this. इसको हटा दो प्रेजेंटेशन को.
सर राशि के पास कुछ प्रॉब्लम हो रहा है उससे।
कोई बात नहीं, आप स्टूडेंट के लिए।
Pratima, you can present.
ओके सर, ओके सर। Good morning, sir. I am Pratima from Bansal Institute of Engineering and Technology, and I am a student of BTech third year. And आज मैं कुछ फार्मर्स की प्रॉब्लम्स वो बहुत सारी हैं, जिसमें से एक प्रॉब्लम को मैंने सॉल्व करने की कोशिश की है। आई एम स्टार्टिंग विद दिस। वंस इन योर लाइफ यू नीड अ डॉक्टर, अ लॉयर, अ पुलिसमैन, बट एवरी डे 3 times a day you need a farmer. So farmers and agriculture, these are very important for us and our country and also our economy. So यहाँ पे जो प्रॉब्लम है, हाउ टू प्रोटेक्ट फार्मर्स क्रॉप फ्रॉम द स्टे एनिमल्स। तो यहाँ पर कुछ फार्मर्स जो हैं वो अपने लोकल ट्रीटमेंट्स यूज करते हैं, लाइक अपने क्रॉप्स के आसपास बैरियर क्रिएट करके, या बीटिंग ड्रम्स, या लाइटिंग फायर्स करके, एंड क्रैकर्स एक्सेट्रा। एंड 2020 एंड 2018 में कुछ साइंटिस्ट ने सम डिवाइसेस क्रिएट किए थे जो कि ब्लूटूथ एंड वाईफाई बेस्ड हैं, एंड it is tough to handle. It is tough to handle for local farmers, and it is very costly. So yes sir,
नहीं, कंटिन्यू प्लीज।
ओके सर। सो या और कुछ बहुत से एरियाज में नेटवर्क इशू रहता है, तो ये डिवाइसेस जो है इतने सक्सेसफुल सभी, आई मीन टू से दैट सभी फार्मर्स ये यूज नहीं कर सकते। तो यहाँ पे मेरा एक छोटा सा सोल्यूशन है, जो कि आई थिंक वी कैन, अगर फ्यूचर में हम लोग यूज करते हैं तो मे बी इसे हम सॉल्व कर सकते हैं। तो हियर इज माय मेथोडोलॉजी। वी in this method we use some eggs and water. Here we used water, normal water, and इसे हम इस अमाउंट में मिक्स कर सकते हैं, लाइक एक एग फॉर 1 liter वाटर। एंड इसे स्प्रे मिक्सचर को बनाने के बाद इसे स्प्रे करने के लिए जो फार्मर्स नॉर्मली स्प्रेयर मशीन का यूज करते हैं, उसे यूज कर सकते हैं। और ये इजी टू हैंडल है फॉर फार्मर्स, एंड ये कोई भी हार्म नहीं करता है, ना ही फार्मर्स को और ना ही एनिमल्स को। तो यहाँ पे कुछ मेरे सम एक्सपेक्टेड आउटकम्स हैं। अगर हम ये इन फ्यूचर इस मेथड को अगर हम यूज करते हैं, तो यहाँ पे जो एग्स का लेफ्ट बाकी बचा हुआ जो पार्ट होता है, वाइट सेल्स, उसे हम एज अ कैल्शियम सोर्स यूज कर सकते हैं फॉर सॉइल की फर्टिलिटी इंक्रीज करने के लिए। एंड ऑल्सो वी ट्राई टू find the particular component which is responsible for producing a bad smell. Yes sir.
ठीक है, बहुत अच्छा। उसकी जो स्मेल से रिपेल करने की बात कर रही हो ना?
Yes sir.
It's an experiment which you said you will also do in your village, isn't it? तुमने अपने अंकल का नाम नहीं बोला जिनसे सुना था ये बात?
Yes sir. So उनका नाम है एडवोकेट शिव कुमार।
और उन्होंने अपने खेत में किया है ये प्रयोग?
Yes sir.
और अब तुम उसको टेस्ट करोगी एक्सपेरिमेंटली कि वो वैलिड है कि नहीं, और उसको फिर बताओगी कि इसको और इम्प्रूव कैसे कर सकते हैं, ठीक है ना?
ओके सर,
बहुत बढ़िया, वेरी गुड,
थैंक यू सर।
नेक्स्ट
Chavi Mishra.
Good morning, sir. I am from Untitled Engineering and Technology. Sir,
Yes,
my
Chavi.
screen is visible. Sir, my
Not
screen
it.
is visible.
Start sharing your screen.
शुरू करो छवि। अगर प्रेजेंटेशन नहीं है तो ऐसे बताओ क्या करने वाली हो।
Sir, my screen is visible.
नहीं, स्क्रीन विजिबल नहीं है बच्चे आपका। नहीं तो आप दिखा दो हमें, अगर आपके पास उसका प्रेजेंटेशन है।
मेरे पास नहीं है।
अच्छा ठीक है, तो
क्या?
हाँ आगे।
Thank you, sir. Sir, मेरा टॉपिक है टू प्रिवेंट खैरा डिसीज विद द हेल्प ऑफ हाइपोमियोकॉर्निया। सर, जैसा कि हम जानते हैं कि इंडिया में बहुत ज़्यादा प्रोड्यूस होता है चावल, तो इसमें एक कॉमन डिसीज होती है जिसका नाम है खैरा डिसीज। तो सर इस थैयर डिसीज कैसे होती है? ये जिंक की डिफिशियंसी से थैयर डिसीज होती है। अब इसका नाम थैयर डिसीज क्यों पड़ा? तो सर एक इंडिया में एक कैचू पाया जाता है जिसको हम खैरा कहते हैं, और इस खैरा जो खैरा डिसीज है इसका कलर बिल्कुल सेम होता है, इसलिए इसको इंडिया में खैरा डिसीज कहते हैं। फिर है सर इसका एक प्लांट पे क्या इफेक्ट पड़ता है? तो सर जो जिंक होता है वो क्लोरोफिल प्रोड्यूस करने में काफी हेल्प करता है। अगर प्लांट में जिंक की डिफिशियंसी होती है तो ये डिसकलर हो जाता है, और जो प्लांट होते हैं वो शॉर्ट हाइटेड हो जाते हैं। और अगर ये डिफिशियंसी है तो जो इसकी बालियां निकलती हैं वो व्हाइट कलर की होती है और उनमें कोई भी सीड नहीं होता है, जिससे हम इसकी इकोनॉमी पे बहुत इफेक्ट पड़ता है। और सर ये है कि इसका ह्यूमन पे क्या इफेक्ट पड़ता है? अगर हम थैयराइड डिसीज लगा हुआ चावल खाते हैं तो हमारी ह्यूमन में भी जिंक की डिफिशियंसी हो सकती है। तो इसमें बहुत सारी प्रॉब्लम होती है, जैसे एलोपीसिया हो जाता है, डायरिया हो जाता है, अचानक से वेट लॉस हो जाता है और भूख कम लगती है। तो ऐसी बहुत सी डिसीज है जो जिंक की कमी से ह्यूमन में हो सकती है। अब इसका ट्रीटमेंट क्या है? तो इस समय तो यूज होता है कि जिंक सल्फेट का यूज होता है थैयराइड डिसीज को ट्रीट करने के लिए। इससे पहले जिंक ऑक्साइड को सीड जो उसके सीड होते हैं उसको ट्रीट किया जाता है। लेकिन पहले के जो फार्मर होते थे वो उसको कैसे ट्रीट करते थे? ये आइपोमिया कॉर्निया नाम का एक प्लांट होता है, उससे ट्रीट करते थे। तो सर ये एक मेथोडोलॉजी है। तो अगर हम इसका यूज करेंगे तो फ्यूचर में इसका यूज कर सकते हैं। सर अगर डिफरेंट डिफरेंट रीजन से सॉइल सैंपल कलेक्ट कर लें, देन उसका हम पीएच, न्यूट्रिशन वैल्यू, मॉइस्चर कंटेंट चेक कर लें, और हर एक जो सॉइल सैंपल है उसकी ट्रिपेट रिएक्शन लगाए अपोमिया कॉर्निया से ट्रीट करके। देन हम उसके सर डिफरेंट डिफरेंट इंटरवल पे हम उसको चेक करते रहें कि उसकी पीएच वैल्यू क्या है, मॉइस्चर कंटेंट क्या है। अगर उन सॉइल पे जिंक का इफेक्ट आता है तो मेंटेन हो जाता है, तो इसका हम प्रोडक्शन चावल मतलब चावल प्रोड्यूस कर सकते हैं, और उसके बाद हम इसको फील्ड ट्रायल पे भी लगा सकते हैं। सर इसके बहुत से इफेक्ट होते हैं कि जिंक की डिफिशियंसी पे अगर हम इसका पीएच है उसको बढ़ा देंगे तो जिंक की अवेलेबिलिटी कम हो जाएगी। अगर फॉस्फोरस का लेवल हाई हो जाएगा तभी भी जिंक की अवेलेबिलिटी कम हो जाएगी, और अगर मॉइस्चर कंटेंट ज्यादा होगा तभी भी जिंक की डिफिशियंसी कम होगी। लेकिन अगर ऑर्गेनिक मैटर को ऐड करते रहें हम तो जिंक की जो अवेलेबिलिटी है वो मेंटेन होती रहेगी। और सर अगर थैयराइड डिसीज लग जाती है हमारे प्लांट में तो इकोनॉमी पे इससे बहुत इफेक्ट पड़ता है।
नहीं, वो तो ठीक बात है, वो तो ठीक है, वो तो दैट इज ऑब्वियस। बस हो गया आपका, और कुछ रह गया?
सर हो गया सर। सर
बहुत
25 से 30% का लॉस होता है। ओके,
थोड़ा
थैंक यू
सा
सर।
एक काम करना बच्चे, एक काम करना, इस प्लांट का एनालिसिस भी करना जो आपने हाइपोमिया करने यूज किया है ना, उसमें
Yes,
क्या
sir.
क्या मिनरल्स हैं, क्या क्या हैं, किस तरीके से वो मोड ऑफ एक्शन क्या है, वो भी स्टडी करना।
Okay, thank you, sir.
बहुत बढ़िया, चलिए नेक्स्ट प्लीज।
युगदर्शनी, युगदर्शनी,
Yes, sir.
yeah, you can start presenting.
So,
शुरू कर दो।
my
बोलना।
topic is to prepare a homemade spray for. So, can you hear me?
कंटिन्यू कंटिन्यू कंटिन्यू।
To prepare a homemade spray for livestock animals against the ectoparasites, sir. So, the original grassroots practices, they have used a dry rigid gauze to scrub on the body of the cattle and give bath to them so that the ticks and lice get paralyzed and fell from the body. So, my idea is that to prepare a bitter gourd seed extract, citrus leaf extract, and a camphor, and that solution will be sprayed on a ectoparasite infected animal in addition to the grassroots practice, sir, so that the ectoparasites will die and the livestock will be protected for next 2 weeks, and that solution will also protect them from bacterial, fungal, and protozoal infections, sir. So the objective is to remove the parasite from the livestock solution, and those solution will also provide them resistant to other bacterial and fungal skin infections, sir. So here is the methodology. I have used hot and cold extraction methods for bitter gourd seed extract and lemon leaf extract, and I also used some camphor. So I have prepared those solution and tested over the ectoparasites. So they were died. I have noted this for 3 days, sir. So after 3 days, they have been died.
Very good.
Thank you, sir.
Very good. Wonderful. So it is in one case, at least we have experimental evidence of some kind, which is good. Now you should have to do it on different animals so that you can do some statistical analysis also.
Yeah, sir. Sure,
Okay.
sir.
Good, good, good. Very good. Next, please.
Thank
Kritika,
you, sir.
How many more, Amar? 1 or 2?
two.
Two bus.
Kritika, Kritika, are you there?
कृतिका, आपकी आवाज नहीं आ रही बच्चे. कृतिका, उसको फोन कर लो, जितने किसी और को बुलाओ जो लास्ट है. अमर, हाँ, कोई जना फोन कर लो हमारे लैब में से और उसको.
Mahi, are you ready?
Kritika, you can call us on phone also. If you want to show your presentation, then you can speak on phone and everybody can hear you.
Mahi, yes.
Go ahead, please. हाँ, बताओ.
Good morning, sir.
Good
Good morning,
morning.
my friends. My name is Mahi Rathore and I'm from Lucknow. I'm a BTech third year student. So this is my topic, role of Heliotropium indicum on insects to protect the vegetables and fruit crops. So this is a fact according to the study of Associated Chamber of Commerce and Industry of India. इंडिया में हर साल 500 बिलियन क्रॉप्स का लॉस होता है जस्ट बिकॉज ऑफ पेस्ट, जो कि काफी इम्पोर्टेंट है. और इंडिया में हर डेली 200 मिलियन लोग भूखे सोते हैं. दैट्स वाई कि प्लांट साइंस इज वेरी इंपॉर्टेंट फॉर अस. सो दीज आर द इंसेक्ट्स दैट अटैक्स द वेजिटेबल्स. आई एम फोकस ऑन फ्ली बीटल्स. सो दिस इज अ फ्ली बीटल्स. एक एडल्ट फ्ली बीटल्स काफी ज्यादा डैमेज कॉज कर सकता है बाय फीडिंग लीव्स एंड स्टेप्स ऑफ द क्रॉप्स. They create a rounded and irregular shapes, usually less than 1 by 8 inches, approximately 0.3 centimeter. These type of damage is very unique for flea beetles. Flea beetles can be black, bronze, bluish, and metallic gray. They have large back legs that they use when they jump one crop to another crop. So इंट्रोडक्शन देने से पहले इससे प्रीवियसली कुछ वर्क हुआ है इंसेक्ट्स को क्रॉप से बचाने के लिए. सो इंडिया में 55% केमिकल इंसेक्टिसाइड यूज होते हैं जिनका काफी नेगेटिव इम्पैक्ट पड़ता है ह्यूमन हेल्थ पे, और जिससे कि हम ह्यूमन को काफी सारी डिसीज हो सकती है. फॉर एग्जांपल डर्मेटोलॉजिकली, रिप्रोडक्टिव एंड रेस्पिरेटरी. और एक एट प्रेजेंट टाइम अकॉर्डिंग टू ICAR, इंडियन काउंसिल ऑफ एग्रीकल्चर रिसर्च, हमारे एट प्रेजेंट टाइम वेजिटेबल्स जो आजकल की हैं उनमें कंटामिनेशन काफी ज्यादा है केमिकल पेस्टिसाइड्स का. फॉर एग्जांपल हरियाणा में 4.5% केमिकल पेस्टिसाइड्स का कंटामिनेशन टॉलरेटिव होता है, बट वहां की वेजिटेबल्स में 56.2% कंटामिनेशन पाया गया है. सो दिस इज माय प्लांट हीलोट्रोपियम इंडिकम, जो कि बिलोंग करता है फैमिली ऑफ बोरागिनेसी. इस प्लांट की जो लीव्स और फ्लावर्स होते हैं वो सन के डायरेक्शन में टर्न होते हैं, और ये इंडिया में काफी सनी लोकेलिटीज, वेस्टलैंड्स पे काफी इजीली तरह से ग्रो कर सकता है. दिस इज माय ऑब्जेक्टिव. प्लांट
कलेक्शन. आप उस पे आ जाओ, अब जो मेथड तो आ गया, आप कैसे मेथड करोगे, मेथड पे आ जाओ.
यस सर. तो ये मेथड है जो मेरी एरिया के किसान यूज करते हैं. ये हीलोट्रोपियम इंडिकम की 2 से 3 प्लांटलेट्स लेते हैं. जिस फील्ड पे ये क्रॉप लगाना चाह रहे हैं स्पेसिफिकली उन्होंने टोमेटो और ब्रिंजल पे ये टेस्ट किया है. उस क्रॉप पे उस क्रॉप को फील्ड पे लगाने से पहले वो इस पेड़ को हीलोट्रोपियम इंडिकम को पहले उसको प्लांटिंग करते हैं. देन उसके बाद जब ये मैच्योर हो जाता है अप टू 2 फीट इन हाइट, उसके बाद वो क्रॉप की प्लांटिंग करते हैं, जिससे कि वहां पे फ्ली बीटल्स के लिए एज एक ट्रैप वर्क करता है. वहां पे फ्ली बीटल्स बिल्कुल भी नहीं आते हैं. दैट्स ऑल.
Very good. So Kritika, you can call me and I will tell everybody your voice, and then you can briefly mention the last minute. Kritika, if you want to try, you can call me and I will share your voice with everybody. आपका mic काम नहीं कर रहा?
Asi, are you ready? If you're ready, you can start.
Good morning, sir, and good morning, my colleagues. My name is Rashi Verma. I'm from the Bansal Institute of Engineering and Technology. I'm BTech student. My research topic on the role of Moringa oleifera for the treatment of bovine mastitis in dairy animals. First of all, I would like to explain what is mastitis. Basically, mastitis is a disease क्योंकि डेयरी एनिमल्स में होती है, तो उनके जो अडल्ट टिश्यूज होते हैं मेमोरी ग्लैंड के, उसमें इन्फ्लेमेशन कॉज होता है ड्यू टू सम फिजिकल इंजरी, ट्रॉमा and some microorganisms named Staphylococcus aureus and Staphylococcus aureus. ये जो बैक्टीरिया है वो फीड कैनाल होता है डेयरी एनिमल में, उसके थ्रू एंटर करते हैं और वहाँ पे इन्फेक्शन कॉज करते हैं, इन्फ्लेमेशन. अकॉर्डिंग टू NCBI survey in Feb 2021, daily losses like for 2 week initial में 1 to 2.5 kg milk जो है वेस्ट होता है, लॉस होता है. And
Asi, you can move to your objective and methodology slide.
Okay, sir. So my objective is first plant extraction and collection. Next is optimized according to the stage of in the stage वो डेयरी एनिमल सफर कर रहा है उसके हिसाब से. फिर उसके बाद हम केमिकल कॉन्स्टेंट के फाइटोकेमिकल हम एनालिसिस कर सकते हैं उनकी कि कौन सा पर्टिकुलर फाइटोकेमिकल है वो रिस्पॉन्सिबल है इस चीज को ठीक करने में, मास्टाइटिस को. एंड हम इसका इफेक्ट देख सकते हैं जो नेचुरल मेडिसिन है इसका. एंड दिस इज माय मेथोडोलॉजी. फर्स्ट हम जो है मोरिंगा ओलेफेरा की कलेक्ट करेंगे, देन उनको स्टेराइल करेंगे वॉश करने से, उसके बाद उनको एक्सट्रैक्ट निकालेंगे. इसमें एक्सट्रैक्ट में हम सब फाइटोकेमिकल कॉन्स्टेंट्स होते हैं, उनको एक्सट्रैक्ट करके उनको एनालिसिस कर सकते हैं, जिससे हमें पर्टिकुलर फाइटोकेमिकल हमें पता चल जाएगा कि ये फाइटोकेमिकल जो है वो रिस्पॉन्सिबल है अगेंस्ट जो माइक्रो ऑर्गेनिज्म कॉज करे इस डिसीज को, उसके अगेंस्ट ये हेल्पफुल है. फिर हम एंटी माइक्रोबियल एक्टिविटी चेक कर सकते हैं मोरिंगा ओलेफेरा की अगेंस्ट द माइक्रो ऑर्गेनिज्म जो रिस्पॉन्सिबल है बोवाइन मास्टाइटिस की. हम इसमें हमारा बेनिफिट ये है, अगर ये सक्सेसफुली एक्सट्रैक्ट हमारा बन जाता है, प्रिपेयर हो जाता है, तो इन फ्यूचर हमारे पास काफी हेल्पफुल ट्रीटमेंट रहेगा अगेंस्ट मास्टाइटिस इन डेयरी एनिमल्स. और सपोज हम पर्टिकुलर फाइटोकेमिकल फाइंड कर सकते हैं, और अगर वो पर्टिकुलर फाइटोकेमिकल लोकल हुआ तो उसको हम जो है पेटेंट कर सकते हैं, और उसका रिसर्च पेपर जो है हम इंटरनेशनल कॉन्फ्रेंस में शो कर सकते हैं.
Thank you, Rashid. Thank you very much. Good effort.
Thank you.
Amar, please. We'll now request the chief guest. ये हटा दीजिए. ये प्रेजेंटेशन हटा दो. हाँ. So friends, we will have the valedictory address now, one for which we were all waiting. It was the brief presentations were just to give an idea as to what kind of experiments that our undergraduate students are going to do. So I will once again welcome Professor Anil Tripathi, Director of Institute of Science, BHU, Banaras Hindu University, Varanasi, former Director, CSIR, CMAP, Central Institute of Medicinal and Aromatic Plant. And in addition to that, he has published extensively. He is J.C. Bose Fellow and received numerous distinctions in his career. He has inspired a very large number of scientists and students to pursue research that is excellent, that is relevant, and that is socially impactful. Professor Tripathi.
Thank you, sir. It's a very special opportunity for me to be invited by you, who has been a source of inspiration to me. And this is because of your selfless dedication to motivate students, particularly in the remote areas, to come forward and contribute in national endeavor of development and building a stronger India. At the outset, I would like to congratulate all the participants who have taken this initiative to get associated with this unique kind of 3-week course, which is giving such a wonderful exposure to all of you to very diverse aspects of biotechnology, and particularly the aspects of biotechnology which have been a kind of gap or missing link in translating the potential of biotechnology in the service of the society. So this course is unique that way. And I congratulate you because in the present times, particularly motivated by parents and society, you like to go for certain disciplines of engineering and medicine. But now, during last few years, this paradigm has changed. And students like you who think differently will certainly distinguish yourself from many others who do not take this kind of initiative and try to understand and explore your capability in different directions. I will give you one example. I heard Bharat Ratna Professor C.N.R. Rao about a couple of years ago. And Professor C.N.R. Rao, he was a BSc student like you. And at that time, he was, there was a lecture of Sir C.V. Raman. And he knew the name of C.V. Raman because he was a Nobel Prize winner. And he decided to go and attend to his lecture. And he said that Sir C.V. Raman was speaking for about an hour. And I did not understand a word of it. But still, I was so amazed by the way he was talking and the all aura around him that kept me inspired. So it is possible that some of the lectures, I have seen the titles, maybe slightly, more complicated, because you need to have basic understanding before assimilating those concepts which were taught to you. But I'm sure that the way the passion with which these experts have explained their work or their vision will certainly infect you, and that will also motivate you to continue further and to find out, to understand what you have not understood. The beauty of this interaction is that you feel that there is something important which you have not understood, but you get ignited to understand those things. And therefore, attending these lectures with full attention is extremely important. So I always remember Professor C.N.R. Rao's statement that I didn't understand a word of it, but still the lecture of Sir C.V. Raman kept hovering in my mind for years to come. And that has kept him motivated. So it's always good that, and I appreciate your effort to attend this course and interact with and listen to many of these speakers. Being the last speaker or speaker during the valedictory function, I run the risk of being repetitive because I do not know that what others have spoken. So it's possible that I might say a few things which might have been said by other speakers. But you should remember that good things should be spoken again and again so that you remember it. They leave permanent imprint on you. The second thing is that being a valedictory speaker, there is expectation that I should be able to make some remarks on the whole course, the lectures which might have been delivered. But since I didn't have that privilege of attending, unlike you, I didn't have the privilege of attending those lectures to make my remarks on those lectures. Now, I would also like to congratulate BIIS and Srishti for starting this program. I can see it's such a rich program. And I said earlier that I feel like attending it myself, because what I study, what I try to research on, that is a narrow area, but it's always good to expose yourself to a wider area if you want to do a sensible biotechnology. Now, biotechnology in India has taken great strides. I think all the lectures which you might have heard, they might have given you confidence and knowledge about how India has moved. In 1986, the biotechnology took birth in this country by the Constitution of National Biotechnology Board of India, and Dr. S. Ramchandran being its first secretary. And since then, it started with the MSc Biotechnology and MTech Biotechnology programs in the country, and it has contributed immensely in developing human resource or manpower well trained in this. But during last couple of decades, the time was not ripe for the harnessing of the capabilities of biotechnology. And there was a lack of atmosphere so that the concepts or the capabilities of biotechnology can be translated to the service of the society. And then came BIIS, the concept of BIIS, that we should somehow connect the science with industry. And that is how this BIIS has promoted a lot of such activities, whereby the people who have some sense of adventureship, they have decided that whatever knowledge in biotechnology is there, that we should try to deploy, to develop some technologies and put it into the practice. And today, whole world is looking at us with surprise that how India has become a world leader in the vaccine program. Today, our capabilities are enormous. And this is because of these initiatives which were taken by our visionary scientists and policy planners a few decades ago. And but there have remained gaps, and these gaps have to be filled. And I think BIIS has taken that initiative, because you might realize that the vaccines which were used, they were produced in India, but their original research was done in UK, for example, the Covishield. So why can't we do the research? We can develop our own patents and produce them in India. That is where we have to be leader in the basic development of the technology. Similarly, when you look at the diagnostics, the government of India had to spend a huge amount of money on procuring these diagnostic kits. And some of these diagnostic kits have been of late developed by the Indian organization. For example, Institute of Genomics and Integrative Biology has developed a diagnostic kit, which is very, very handy. But World Health Organization recommended diagnostic kit turned out to be very costly, because our population is huge and we have to find newer solutions, cheaper solutions. And it is people like you who have to feel such kind of challenges, that yes, we have to find a way out so that the amount of money spent on one diagnosis, we should be able to do 10. And I think the solutions were done, the group testing was done, and that is how the price were economized, and such a huge testing of COVID infections were done in the country. So there was a gap, because the raw material, the enzymes and other material used in diagnostic kits that were all imported from outside countries. So there is a need felt that we should be able to produce it ourselves, so that we become self-reliant. We should become Atmanirbhar. So that attempt is being initiated by BIIS to identify the persons with the entrepreneurial spirit, and so that they have the guts and determination to take the concept, knowledge, and technology into the form of the formulations which can reach to the society. And you know that most of the enzymes which are needed in the country for textiles, for leather, for many, many other applications, most of these enzymes are imported by India. Many of us can take up that we should be able to produce this enzyme. We should develop state-of-art technology for the production of enzyme indigenously in India. I'm reminded of one visit of Dr. M.K. Bhan to Banaras Hindu University, and I was accompanying him. And there is my senior, he was telling that the lot of research, the lot of money is going to the basic science, to the biotechnology, and actually it should go to life science. And then Dr. Bhan visited the departments of zoology and botany, and then he came to biotechnology. To all these students, he asked a question that what do you want to do in future? The students in life sciences said that they want to understand this, they want to understand that phenomena like that. But the biotechnology student said that we want to produce a monoclonal antibody. I want to produce a specific enzyme. I want to produce a specific transgenic plant. So there is the difference between life science and biotechnology. In life science, you try to understand the phenomena. In biotechnology, you try to deploy that knowledge for addressing some problems and mitigating the problems of the society. So that is the difference between, that's how Dr. Bhan showed it to our colleague in life science that, see, this is the difference between life science and biotechnology. And today, when you are all taking this training or attending this training program, I'm very glad that many of you have already started looking at your presentations. You have already started thinking of a problem. There are two methods of doing science. One is that you raise a very basic question and try to use all methods to address this fundamental question. The second way of doing science is you identify a problem and use all knowledge available to address that problem. And I think there our country has been lacking. And this course has a special significance that you have to learn to deploy the knowledge. And that is why you have been exposed to a wide variety of domains of knowledge so that you are able to connect it in future. As I said, that today is the right time to take up biotechnology as a career. About 20, 30 years, it was not so. Because I again am reminded of one instance that one mother, she came to my office and said that my son is getting admission in civil engineering in some NIT or so, and he is getting admission in biotechnology in some good IIT Delhi or so. So at that time, about I think 15, 20 years, when that mother came to me, I told her that you look around in this room, around your ambience, that where is the civil engineering and where is biotechnology, whether any product of civil engineering is there. So these walls and these toilets, everything is civil engineering, and everyone needs a house to live. And therefore, there is a demand for a civil engineer. But where is the demand for biotechnologist? A small child, a small baby goes to the doctor for some kind of vaccine. Maybe there is some kind of biotechnology, but otherwise there was not much of biotechnology needed in the society. But today, during last 2-3 years, courtesy pandemic, this COVID pandemic, everybody talks about biotechnology because existence of life is important, and existence of life depends on the diagnosis of the disease and cure of the disease and prevention of the disease. And therefore, the importance of biotechnology has suddenly shot up these days, and you are at the right time, at the right juncture to pursue your interest in biotechnology. Earlier, the transgenic technology, developing transgenic plants to address the problems of food and nutrition, this had developed with a great pace. But because of the restrictions on genetically engineered organisms, this could not find place in practice. In India, except for BT cotton, we do not have permission for any transgenic crop. Whereas many other countries, including China, Bangladesh, they have allowed these transgenic crops. And I think this, with new advent of technology of the genome editing, genome engineering, now it should be possible to make the desired changes in the plant, and they should be acceptable. They should be much better and risk-free as compared to the transgenic plants. Now I will ask you one question that I always think that why should Professor Gupta organize such a course? What is his motive to organize this course? And when I sort of try to contemplate, I feel that he is trying to secure the future of India through empowering our youth, empowering our generation with the knowledge, and by instilling their confidence in the traditional knowledge so that they explore it and try to find the potential. Because today, every medicine what we have, almost every medicine what we have, this is based on traditional knowledge about the plants. Even the aspirin which you have, this aspirin, idea of aspirin came from a willow plant. And later on, when the compound was identified, it could be industrially synthesized. A simple compound could be synthesized. So the most commonly used medicine, the idea came from the plant. And therefore, the idea is that the traditional knowledge has a lot of reservoir of knowledge which needs to be picked up, validated, and put into practice. And the other point which I see that Professor Gupta is trying to make the country's future secure, he carries out the padyatras. I do not know whether he mentioned about his padyatras. And these padyatras are basically to make you aware of the ground realities of the country. Unless if you remain restricted within a lab, you don't come to know what are the actual problems of the country and what are the different phenomena which are present in the nature. And unless you go into the lap of the nature, whether it's a harsh summer or a raining period, when you go to the close to the nature, then you realize that what mysteries of nature are there, and you get to know so many questions. For example, I will just quote one example, that in my morning walks a few weeks back, maybe still going on, it's a leaf fall. The tall trees are there and the leaves are falling. And I see every day that every leaf falls in a different way. Its pattern of falling is different. And this is different because of the size of, shape of the leaf, the way it is twisted. And according to that, it keeps on, it falls in a different way. And I was thinking that maybe we should try to find a correlation. There are methods these days to record it and look at the shape of the leaf and what is the, it's a trajectory, how it is moving. So these are when you see the nature always ignites you, always floats some questions which you like that, yes, you should try to address it. And I think this is very important that we all should go close to the nature. Every scientist in the world who has done outstanding observations, outstanding research, they had the habit of going close to the nature. Be it Sir C.V. Raman. Sir C.V. Raman, when he's on the ship, he was asking the question that why the sky is blue and the ocean is green. And similarly, he used to ask many questions that why a specific musical instrument gives a specific kind of sound. These are all nature. You try to understand that how does nature behave, and that brings you Nobel Prize. Not one, multiple. All those who have got Nobel Prize, they have always been trying to explore and investigate that how does nature behave. I will give you many more examples if time permits. But what I'm trying to say that this grassroots innovations which Professor Gupta is trying to highlight, and for which he is known, he has dedicated his life. I think a lot of wisdom is hidden there, and we have to see that we have to translate that wisdom into a widely accepted practice, and that will happen by persons like you if you devote yourself to find the science behind it, logic behind it, so that it becomes acceptable by all. And that is where you need to do reverse engineering. Reverse engineering, one thing is engineering. When you put science into practice, then it is engineering. When you understand science behind a practice, then it's a reverse engineering. And I think these age-old practices of traditional formulations, they are there. We know the cause and effect relationship, that if you do this, something happens, something positive happens. That why does it happen? That is what we need to understand. And therefore, it is important to sensitize and ignite young minds about your capability in addressing problems facing our society. I gave you example of Sir C.V. Raman. Now, I'm sure that you know the names of Charles Darwin. You know the name of Gregor Mendel. Gregor Mendel was a priest, and he was looking at nature, that why pea plants are giving a specific kind of variation, and he tries to apply some kind of mathematics. Although he was not a student of mathematics, but he applied mathematics and he drew certain laws for his own satisfaction. This was only watching in nature, observing the nature very keenly, that how does nature behave. Similarly, Charles Darwin took up a ship and went around the world to find out that how different life forms are related with each other. And then he proposed the origin of species, the theory of evolution. And these are the phenomenal people whom we always remember, and we always owe for the present developments in science. And therefore, nature is a greatest of all teachers. And therefore, all of us should learn from Professor Gupta's example, that we should always make it a point to go close to the nature, observe the nature without disturbance, without talking to your friends, just observe the nature, and always you will be able to find some new problems, new issues, which will give you a lot of name, fame, and satisfaction. Now, one thing is the creation of knowledge, and second thing is deployment of knowledge. Life science is creation of knowledge, and biotechnology is basically, if a technology doesn't get deployed, it doesn't serve the purpose. So we have to solve all the issues till a particular biotechnology reaches to the society. And therefore, innovation is basically deployment of knowledge. So you create knowledge. Every scientific institution or educational institutions have three mandates. One is the creation of knowledge, second is dissemination of knowledge, and third is deployment of knowledge. So in life sciences, we create knowledge, we disseminate knowledge, but in biotechnology, we deploy the knowledge. But for doing biotechnology, for putting biotechnology into practice, you need to understand different dimensions of knowledge. And that is why you need to understand physics, chemistry, computer science, mathematics, mechanical engineering, chemical engineering, nanotechnology, and several others. Because everything is important and everything you will have to integrate. I will give you another example, that few years back, I had gone to Czechoslovakia, its capital, Prague. And in Prague, I met a girl. She was doing PhD in mechanical engineering. And she was an Indian. So I asked her that, what is your background? She said, I did MSc Zoology. I said, how come a MSc Zoology student is doing her PhD in mechanical engineering? Then she said that in this institute in Prague, my knowledge, my familiarity about the birds is being used for developing flying machines, developing certain principles for flying machines. And I was really amazed that this is a kind of interdisciplinary interaction which is to be utilized in creating new kind of applications. So the birds, if you look at the sky, you find variety of kind of birds. And these birds have different ways of flying, floating, hovering, and there are different mechanisms. Muscular mechanism, musculatures are different, their blood supply is different. And all this is having specific significance in developing variety of kind of machines. When you look at the airplanes or helicopters or choppers or even the drones, we apply only a few mechanisms of flying which are there in the birds or in the insects. But there are many such mechanisms which need to be applied. Later on, when I started discovering, I found that our great Sir Satish Dhawan, he had written a monograph on the flight mechanisms of birds. And because he was a space scientist, he had already tried to observe the nature and try to apply those principles for space research and for the flight related machines. Now I will give you a few examples that what is biotechnology and what it requires to be a biotechnologist. Now I will try to give you a few examples with which you might be familiar. You see, biotechnology requires integration of knowledge. And this integration of knowledge, for integration of knowledge, you should have, first of all, a good exposure, good basics of diverse disciplines of knowledge. You might have, I'm sure you might have read about the development of the double helix model of DNA. You have been studying since your school, high school or intermediate, you might have heard about it. Now, if you ask a question that what was the contribution of Watson and Crick? Watson and Crick did only integration of pre-existing knowledge. They did not do any experiment. They only, studied everything and they put everything together like a puzzle, like a Lego, construction. All the things they put together, and eventually a double helix model came, which has revolutionized molecular biology and biotechnology. Whole development took place by that. But you know what is the genesis of that? Watson and Crick both studied a book written by Erwin Schrödinger, which is called What is Life? Erwin Schrödinger in 1940s, he wrote a book, What is Life? And many of the Nobel laureates in the future years, they studied this book and they got the idea that they should try to understand what is that basic molecule which is responsible for life. And Watson and Crick, they got the credit for it, including Wilkins, he got a Nobel Prize. But Watson was a biologist. He used to study viruses. Crick was a physicist. He had actually participated in the atomic bomb project, and he was highly disillusioned by this. And therefore, he decided to go to biology, but he was a physics person. Now, Rosalind Franklin and Wilkins, these all were physicists. And physics and biology people, they came together trying to understand the structure of a molecule which is responsible for life. And on the other hand, there was a fusion of chemistry. Erwin Chargaff, he gave the basic rules of the base pairing. And Linus Pauling, he was also a chemist, and he gave the ideas of making these kind of helical structures. So when all these people came together, I mean, if you read about the life history of Watson and Crick, they stole at times the ideas, particularly from Rosalind Franklin, and they put it together to make the model. And still they were not able to make a model. And on 28th of February, they were able to see the model falling in place. And one day before that, there was a scientist, Jerry Donahue. He gave them the idea that the backbone should be on the outside and bases should be inside. Earlier, Watson and Crick were putting backbone inside and bases floating outside. But Donahue said that, see, the chemistry doesn't work that way. The bases have to be inside. And then the double helix model fell in place. Now, who should get the Nobel Prize? Donahue, who gave the final trigger because of which the model fell in place. No, I mean, we should not go into that controversy. What I mean is that Watson and Crick, they were the anchor persons behind the construct of the double helical model of DNA. There were contributions from several disciplines. If they had not understood these things well, they would not come up with a double helical model. I'll give you another example. So a biotechnologist need to be a person who integrates knowledge. To solve a problem, he or she collects information from different domains of knowledge and puts it together to address that question. The second example which I would give is about Cary-Mullis, the polymerase chain reaction, which you might have heard about. The polymerase chain reaction was originally an idea of Dr. Hargobind Khurana, who thought that DNA can be replicated into two molecules when you give a primer and some an enzyme, DNA polymerase enzyme, and this can be replicated. And they gave a concept of this polymerase chain reaction in the beginning. But this was only a concept. This was not experimentally demonstrated. Cary-Mullis was a scientist in the Cetus Corporation, a company, and he was doing the experiment, the same kind of experiment. He was designing the primers and trying to extend them and make the synthesis. But always he used to think about this experiment, and he got the idea that probably it is possible that we should be able to make multiple copies of the same DNA if we are able to use polymerase again and again. And he did not do it, but his colleagues in the lab, they were able to demonstrate that the idea of Cary-Mullis is right, that if we are able to provide DNA polymerase every cycle, because you denature and then at 95 degrees to denature the DNA, then again provide primers and give the enzyme. So again and again you have to provide the enzyme in order to carry out the polymerase chain reaction. Now look at that. This is what was the concentration, the idea of Cary-Mullis. But it became feasible by a Thomas Brock, a scientist, a microbiologist, who had very weird kind of interest. He wanted to know whether life exists at very high temperature or not. And then he was exploring the microbes present in the thermal spring, Yellowstone thermal spring. And then he isolated various bacteria, including Thermus aquaticus. And this Thermus aquaticus, he didn't know that what is the potential of Thermus aquaticus, except that it can tolerate up to 80-85 degrees Celsius and can survive. That was the only thing which he knew. And he was going to close his research. Then another biochemist, he picked up this research and he isolated variety of kind of enzymes. And he found that there is a DNA polymerase which can act at 80 degrees or 85 degrees. Now these ideas, random ideas, they were all picked together, and the success of this polymerase chain reaction started when this thermostable Taq polymerase was put into practice. And then it was possible, but this polymerase chain reaction started working, but polymerase chain reaction was not very handy, because every time you had to shift the reaction from one temperature to another, and that's why you have to keep on rotating the temperature. And then some mechanical engineer had to be, the help of mechanical engineer had to be sought. And then he made a thermal cycler, which can increase or decrease the temperature at a very high rate. So you don't have to shift your vials, but you keep at one place and the temperature can go up and down. And that is how that became the beginning of the success of this whole phenomena of polymerase chain reaction. The contribution of Cary-Mullis was very limited in this. Cary-Mullis did not discover the enzyme. The concept was Hargobind Khurana, and this extension of DNA, that was Arthur Kornberg. He showed that if you have a DNA and you have a primer, it can be extended. But he was trying to put everything together. Cary-Mullis was trying to put everything together. He did not put everything together. Somebody else did it, but Cary-Mullis got the Nobel Prize. There were so many controversies about it. Many people asked that did he deserve? Because he was a very passionate scientist. He always was thinking about it, although he did not do it, demonstrate it himself, but he got the credit. And he was a very weird kind of scientist, very addict of LSD and marijuana and those kind of things. If you read the character of Cary-Mullis, you will be highly, disgusted by his personality. But one, there are two important features of his personality. One is passion. He always was thinking whether he is under effect of marijuana or he's drunk with the alcohol. He's always thinking about one question. And the second thing is to put the things together, to integrate everything together. That was the material, and that is a most revolutionizing discovery of biotechnology. And today you know that what is the revenue earned out of the production of Taq polymerase. Because every lab, every technology of molecular biology is based on polymerase chain reaction. But look at just the beginning, that a person tried to integrate everything together. I'll give you a couple of more examples because it's very, inspiring to see that how biotechnology has developed and what are the main features of a biotechnologist which actually matter. Now, interaction and collaboration, these are extremely important features for the development of biotechnology. You have to be open to ideas. You have to seek out ideas. You have to interact together and put the two kind of ideas together. And the first recombinant DNA, that is called the birth of biotechnology, the creation of the recombinant DNA molecule. This is because of coming together of two persons. One was interested in viruses, another was interested in antibiotic resistance, mechanism of antibiotic resistance. The one who was interested in viruses was Herb Bayer at the University of California, San Francisco. He was expert in restriction enzymes. And actually, he has carried forward this legacy from Werner Arber, who was actually awarded Nobel Prize for discovery of the restriction enzyme. Now, look at the basis of restriction enzyme. Where from you get restriction enzyme? E. coli, any alien DNA, whether it's a bacteriophage or a plasmid, tries to enter inside it, it has a mechanism to restrict it. And that mechanism is a scissor. In a gross language, you call it as a scissor, otherwise it is an enzyme. So anything which is alien to it, it chops it out, it cleaves it, and therefore it cannot survive and it cannot invade. That is the enzyme, restriction enzyme, that has revolutionized or actually was responsible for the birth of biotechnology. Now the Herb Bayer, he was working at UCSF, and in that area you call Bay Area, at University of Berkeley, there was another scientist called Stanley Cohen. And Stanley Cohen was trying to understand the molecular basis of antibiotic resistance. And then he came to know that there are some extra chromosomal elements which are called plasmids, which are responsible for conferring this antibiotic resistance. And then they met in a conference in Honolulu at Hawaii, island. And then they were sitting on a beer table, and they were talking to each other, and they suddenly got inspiration to learn from each other that if they work together, they can do some good useful experiment. So Stanley Cohen said, okay, I can offer you the plasmids which I have. And Herb Bayer said that, okay, we have restriction enzymes, we can try to do this experiment together. And then one of the technicians shuttled between the two labs, and then they were able to create the first recombinant DNA by applying restriction enzyme on the plasmids which were there with him. I will not go into the details, but understanding these basics are important in order to be a successful biotechnologist. So the lesson here is that you should be open to discussion. Always discuss with a scientist who works in a different area and try to understand what he or she is trying to do, and then try to learn, take home some lessons and try to apply it with you. And you should be ready to also share your ideas, because unless you share your ideas, the other person would not be able to appreciate and come forward for collaboration. And therefore, I think sharing ideas, interacting with each other freely, and collaborating is the crux of biotechnology. Another example, which I would like to give you, is that biotechnology requires a deeper understanding of biology. You are undergraduate students, you should see that you understand, try to go deeper into the phenomena. And I would like to give you an example of the transgenic technology. So many transgenic plants have been developed all around the world with variety of kind of traits which were not possible earlier. How it became possible? This became possible by the effort of few plant pathologists who were looking at the junction of root and stem, where some crown, this location is called crown, some gall kind of structures, they were formed, and they were trying to know what is the basis for this crown gall formation. What is this pathogenesis, mechanism of pathogenesis? And when they went deeper into it, there was Mary Chilton in US, Von Montague and Jeff Schell in Belgium. They were working on these aspects. And then they realized that there is one bacterium which is so intelligent that this is able to deliver a piece of DNA into the host plant, host cell, where it gets integrated. And then it makes the host plant as a slave. It makes the cell of the host plant as a slave, and the plant starts doing what this bacterium wants it to do. So this was a natural genetic engineer, Agrobacterium tumefaciens. This is the bacterium. This knows how to make the plant as its slave. What we did, what molecular biologists did, to understand how does it do this trick. And once you know this trick, you know how to engineer the plant and deliver the genes of your own choice into the plant. And that is how this transgenic technology developed. But this would not have developed because of the collaboration of these three scientists, but also a company called Monsanto that came into the being, because Monsanto was using the pesticide. It was a company which was producing pesticide and distributing pesticides to the farmers without realizing that how much havoc it is creating for the health of everyone. And later on, when the Raquel Carson, she wrote a book on Silent Spring, then people became aware that how much damage is being done by the pesticides. And then Monsanto also became aware, and their existence came into the question. So they started investing heavily on these three scientists, that can we develop some technology? And that is how that today the BT cotton, a variety of BT crops are there, and they save a lot of pesticide use because the plants are resistant. So so another important aspect of biotechnology is that you should be open to interaction. You should be able to attract industry so that they can invest. Otherwise, science cannot grow further. So industrial partnerships are important. To a certain extent, the government can fund your ideas, but your ideas have to mature to a level that you are able to attract funding from other sources which are in the business-like mode. So to sum it up, I would like to say a few points that what are the essential ingredients of a good biotechnologist. Number one, as I said, that there has to be urge, passion, curiosity to address mysteries of nature. But these are the features of a biotechnologist, of a biologist. But the unique features of biotechnologists are to integrate the knowledge, to deploy the knowledge, to understand the knowledge, its usefulness, and put them together across disciplines to address some existing challenges. That is the uniqueness of a biotechnologist. And then you should have a broad heart, and a good, personality to interact with the people. You should be open enough so that everybody comes to you. You can reach out to everyone. You should be able to have collaboration. And whether it is research or for biotechnology or for any major mission or venture, you need to have perseverance and patience. You should be open to ideas and suggestions, as I told you about the creation of recombinant DNA. And there has to be a continuous process of learning and updating yourself. Because today you develop a technology, and tomorrow your rival is again coming up with another technology. And your technology will become obsolete after a while. And therefore, this biotechnology being or sustaining or surviving in biotechnology requires continuous upgradation of your knowledge, putting up always new ideas, finding new ideas, and putting them into practice. So the continuous learning and updating has to be your characteristic of your personality. And then another most important thing is to have the passion to take your invention or your innovation to solve problems to the society. This is most important thing, because a lot of knowledge have been generated, but unless that knowledge reaches to the society, it doesn't serve the purpose. Because we should always think, when we are generating knowledge as a scientist, remember the 7 sins which Gandhiji mentioned once in Young India article. And one of the sins, very important sins, is science without humanity. If you only care about your curiosity, that is not enough. You have to see that the resources which are provided to you, they are of some use to the humanity. So science without humanity is a very, one of the 7 sins which Gandhiji mentioned about it. So I think we should always remember that whatever effort we make, whatever the Government of India programs, BIIS programs, the efforts of Professor Anil Gupta, all these will come to fruition when some of you will take up some of the existing problems of the country. There are some knowledge base available. You make use of that knowledge base through the grassroots innovations. Try to validate that, have your own new ideas, try to develop your ideas to the extent that industry comes to you to partner with yourself, so that your idea gets deployed for the benefit of the society. So I think I have taken a long time, but I thought, teachers usually take longer. I have been a teacher of biotechnology, and I thought some of the basic things I will try to put into perspective, so that that what are the major discoveries. I have not mentioned, discussed many other things like genome editing and many other things, which are again the urge for understanding the basic biology. Because of that, all these things have come out. So I think you should maintain basic curiosity, but also urge for applying all this. And today's, this 3-week course prepares you for that, because you are under undergraduate. You are already exposed to some of the ideas, how to put your idea into practice for the benefit of the society. Now, when you go back and study your regular studies, you have to know that what are the things you have to focus on, and what are the realities, ground realities of being a successful biotechnologist or a scientist in general. So I sincerely thank Professor Gupta for giving me this opportunity to share my excitement about science and biotechnology and biology, and also to express that how much I admire his efforts to prepare the younger generation to take on the future challenges to make our India self-reliant and Atmanirbhar. Thank you so much, sir.
Thank you so much, Professor Anil Tripathi. And I can tell you one more thing, the thought which came to my mind. In the next BIIS, we will begin it, the formal part of the course, with a lecture from you. An introductory lecture. It was two differs, the whole genesis of how the field has evolved over the years with different scholars and different controversies that have arisen in the process. It's very important for students to know that brick by brick, the structure is made, but a lot of people make different bricks, and they all come together at some stage by different people. And then again, the bricks start falling apart, and somebody again brings them together. So it is a process of a lot of people contributing to a tree of knowledge. And I think you brought this out very well. You also brought out some other aspect of the knowledge, how to know. And the example that you gave of a leaf falling and in the morning walk that you observed is a profound insight that all around us, there are questions waiting to be analyzed, things waiting to be understood. And the fact that the different leaves, because of the curling that takes place in the process of drying, allow the friction to be navigated differently. Some fall on the road and some fall in the soil. All of that makes a difference to their destinies. There is a very old poem, I think, of Maithili Sharan Gupta, probably, that a drop of rain, where does it fall? Does it fall in a sippy to become a pearl, or does it fall to quench the thirst? And I was reminded of that poem when I heard your description of that leaf, that there are multiple destinies that these leaves which are falling. And since some of our students are looking at the analysis, they would also now have to ask a question: which leaf? Young leaf, old leaf, dried leaf, newly sprouted leaf? All of them may have different properties. So I think you have sharpened their observation, their perception. And I also think that our own team in Srishti will benefit a great deal from this interaction. So I thank you once again for a very insightful, very powerful lecture, which I'm sure will stay with all of us, but particularly with the students for a long time. As you mentioned about the Senior Rao's example, that many times if students don't understand something, doesn't mean that they have to understand it then and there. The unanswered questions must be kept in mind so that at an appropriate stage when mind is ready, the question comes up and then gets resolved. So that's very kind, very nice of you. My old genetics of 1970s got reminded today. I have 1 degree in biochemical genetics, and that's how my interest continues to get sustained when I hear such deeply illuminating lecture. So thank you so much, sir. Amar, the vote of thanks.
Thank you so much, Professor Tripathi. I invite Chetan Patel to deliver the vote of thanks. Chetan?
Yes. नमस्कार. हमारे जो 20 के बच्चों के लिए अनिल सर त्रिपाठी ने जो अपना वैल्यूएबल समय निकाला, उनके लिए मैं सृष्टि के तरफ से मैं उनका आभारी हूँ. बेसिकली ये जो बच्चे हैं वो बड़ी इंस्टीट्यूट से नहीं है, IITs, NITs ऐसे कोई इंस्टीट्यूट से नहीं है, छोटी इंस्टीट्यूट से आए हैं. और 20 के दौरान काफी सारे ऐसे लेक्चर उनके साथ हुए कि भविष्य में शायद उनको ऐसा मौका मिले ना मिले. तो सभी जो गेस्ट है उनका मैं आभारी हूँ कि जो 20 जो हमारे 21 दिन का प्रोग्राम था उनमें उन्होंने अपना जो टाइम है निकाला. दूसरी चीज के 20 के माध्यम से जो हम सालों से जो बात कर रहे हैं, जो फॉर्मल साइंस और इनफॉर्मल साइंस के जो हमारा घर का जो विज्ञान है उनके साथ हमारा जो विज्ञान है, जो हमारा साइंटिफिक विज्ञान है, उनको हम जोड़ने का काम नहीं कर रहे थे. लेकिन जो हमारे होम एक्सपेरिमेंट के वजह से बच्चे अपने जो वडीलों के पास जो नॉलेज था उनके अंदर भी साइंस ढूंढने का काम किया. तो एक अच्छा जो अचीवमेंट जो है वो हमारा बन सकता है. और जो हमारी न्यू एजुकेशन पॉलिसी है उसमें ये सारी चीजें का ऊपर ध्यान दिया है, लेकिन उससे पहले हमने उनको शुरू कर दिया है. और दूसरी चीज के जितने भी लोग आए उनका भी मैं आभारी हूँ, सभी बच्चों का भी आभारी हूँ कि उन्होंने भी जितने भी हमने असाइनमेंट दिए उनको अच्छे तरह से उनको काम किया. घर में रह के अभी तक हम सोच ही रहे थे कि अच्छे एक्सपेरिमेंट लैब में होते हैं, लेकिन कुछ बच्चों ने हमें सिखाया कि घर में भी अच्छे एक्सपेरिमेंट कर सकते हैं, कम रिसोर्स में, कम जगह में. तो एक अच्छी अपॉर्चुनिटी भी बच्चों ने खड़ी की है कि भाई इस तरह का भी एक नया माहौल क्रिएट हो सकता है. दूसरा काम के 20 के दौरान जितने भी हमारे जो लेक्चरर्स थे उन्होंने जामवंत का काम किया है कि जो हमारे बच्चों के अंदर जो पहाड़ को ले जाने की, समुंदर को पार करने की जो बच्चों के अंदर ताकत थी उनको उजागर किया है, यानी कि रिसर्च करने की उनकी एबिलिटी थी उनको बढ़ाया है. तो मुझे लगता है कि ये जो बच्चे हैं आगे जाके अच्छे रिसर्चर बनेंगे. और जो हमारे जो परंपरा रही है कि हम हमारा जो सामान है वो टन के दाम में बेचते हैं और मिलीग्राम के दाम में हम खरीदते हैं, उसके जगह ये बच्चे हैं वो टन के दाम में नहीं लेकिन मिलीग्राम के दाम पे बेचेंगे, ऐसी मुझे पूरी उम्मीद है. और सभी बच्चों का, पूरे हमारे स्टाफ का, जो बायरैक की टीम का, सभी का मैं इस समय पे आभारी हूँ कि ये जो बढ़िया आयोजन में सभी ने सहयोग किया. अमर का भी, क्योंकि अमर ने डे नाइट्स इस काम में लगाया है, कम स्टाफ की वजह से भी उन्होंने, और बाकी सारे स्टूडेंट के जो अमर को बैक में जो क्विज बनाने में सारा काम में मदद की, तो उनका भी आभारी हूँ. बाकी हमारे फील्ड स्टाफ, बाकी सभी का इस वक्त पे आभारी हूँ. और उम्मीद है कि आप सब लोग हमसे जुड़ते रहेंगे और जो आपने जो रिसर्च का जो काम है वो सृष्टि लैब के साथ मिलकर हम उनको आगे बढ़ेंगे. थैंक यू, धन्यवाद.
Thank you, Dr. Tripathi. Thank you, all the participants. Please stay in touch, finish your assignments, and I'm hopeful that we will hear about your achievements in future. Thank you, Professor Tripathi, once again.
Right.
Thank you so much.
Thank you, sir, and my best
Thank you.
wishes to all the students who have participated in this course. All
Thank
the
you.
best.
Thank you, Professor.
Thank you.
Thank you all for joining us today. Thank you.
अब खत्म करेंगे ना?
हाँ
जरा खत्म हो जाए फिर कॉल कर मुझे।
हाँ